Good morning, everyone. Today, I'm going to be talking about one of the most important molecular mechanisms that allows cells to control which genes are turned on or off, which is DNA methylation. This process is central to the field of epigenetics, which is the study of heritable changes in gene expression that occur without altering the DNA sequence itself. By definition, methylation is the process in which methyl groups, also known as ROCks, are added or removed. Epigenetics can be thought of as a layer of regulation on top of the genome. Every cell in your body contains the same DNA sequence, but epigenetic marks determine which genes each cell actually uses. This is why a neuron and a skin cell can look and function completely different despite having identical DNA. A major way this happens is through chemical tags that decorate DNA or the proteins associated with them. One of the most fundamental of these tags is DNA methylation. What is a methyl group? Methyl groups are simply carbons with 3 hydrogens attached to them, as displayed chemically on the screen. This methyl group is usually added to the 5th carbon of cytosine bases in the DNA sequence, typically in regions where cytosine is followed by guanine, known as CpG sites. This process of DNA modification is performed in a reversible manner. This reaction is carried out by a family of enzymes called DNA methyltransferases, also known as DNMTs. When a methyl group is added, it doesn't change the letter of the genetic code, but it can change how the gene behaves. In general, DNA methylation is associated with reduced gene expression. Heavily methylated regions of DNA tend to be tightly packed and inaccessible to transcription machinery. You can think of methylation like placing a closed sign on a gene. The gene is still there, but the cell is less likely to read it. As a result, the gene becomes inactive because the transcription factors cannot bind to the DNA any longer and are being blocked. Therefore, there is no gene expression occurring, also known as transcriptional repression, essentially turning the gene off. However, methylation isn't always a simple on or off switch. Sometimes it fine-tunes expression or ensures proper timing and tissue-specific control. One of the most important roles of methylation is gene silencing, which includes shutting down transposable elements, which are jumping genes that can be harmful if left active, silencing imprinted genes where only one copy, maternal or paternal, should be expressed, and inactivating the second X chromosome in females, forming the Barr body. These examples highlight how methylation protects the genome and maintains correct gene dosage. When cells divide, they need to copy not only the DNA sequence, but also the methylation pattern. This is done by maintenance methyltransferases, specifically DNMT1, which recognize methylated cytosines on the parent strand and place methyl groups in the equivalent spots on the new strand. This means methylation patterns can be inherited through cell divisions, allowing cells to maintain their identity over time. DNMT1 appears to be responsible for the maintenance of established patterns of DNA methylation, while DNMT3A and 3B seem to mediate establishment of new DNA methylation patterns. Diseased cells, such as cancer cells, may be different in that DNMT1 alone is not responsible for maintaining normal gene hypermethylation, an increase in global DNA methylation, and both DNMTs 1 and 3B may cooperate for this function. One of the most fascinating aspects of DNA methylation is that it is influenced by the environment. Factors that can alter methylation patterns include nutrition, stress, physical activity, exposure to toxins, and aging. For example, identical twins who share the same DNA become more epigenetically different as they age, partly because of changes in methylation patterns. This illustrates that while our DNA sequence is fixed, our epigenome remains dynamic and responsive to life experiences. A specific example of this mechanism is heterochromatic methylation. This process is effective at maintaining chromosome stability by preventing translocations and other chromosomal abnormalities. Heterochromatin is a fundamental architectural feature of eukaryotic chromosomes that endows particular genomic regions with specific functional properties. The term heterochromatin was coined based on the differential staining of chromosomal regions, but now generally refers to molecular subtypes of transcriptionally repressed chromatin domains that extend beyond a single gene or regulatory element. Because of its importance for chromosome function, heterochromatin has key roles in the pathogenesis of various human diseases. The methylation of H3K9 is crucial for the stability of heterochromatin and is recognized by the chromodomain of HP1 proteins, which recruit other chromatin-modifying enzymes to maintain the heterochromatic state. The bulk of methylated CpG dinucleotides are found in the repetitive DNA sequences located in the heterochromatic regions of the genome, including the centromere. Histone PTMs regulate the propensity of the underlying DNA to participate in the process of transcription, replication, repair, and recombination. Specific PTMs control binding of particular proteins to nucleosomes via specific protein domains known as reader domains or modules. This dynamic process requires the continuous action of multiple chromatin-modifying enzymes and is essential for regulating gene expression by silencing or repressing gene activity. Given its role in disease, scientists have developed therapies that target methylation. Some drugs, such as azacitidine and decitabine, inhibit DNA methyltransferases and are used to treat leukemia and other cancers. By reducing abnormal methylation, they help reactivate silenced tumor suppressor genes. This area of epigenetic therapy is one of the fastest growing fields in modern medicine, offering a way to potentially rewrite harmful epigenetic marks. To summarize, DNA methylation is a key mechanism of epigenetic regulation that influences gene expression without changing the DNA sequence. It plays essential roles in development, cellular identity, and genome stability. Environmental factors can alter methylation patterns across the lifespan, and abnormal methylation is associated with a wide range of diseases, including cancer. DNA methylation acts as a molecular switch that helps cells interpret the genome. It is central to how organisms develop, maintain health, and respond to their environment. Epigenetics, and methylation in particular, remind us that our biology is written not only in the letters of our DNA, but also in how our cells choose to read those letters. Thank you, and here are my sources.